Okay, we are going to go ahead and get started. This is Holly Ferning with the University of Illinois Extension. I am the Master Gardener and Master Naturalist Coordinator, and this is our third in our series of Wilderness Wednesdays. And we are so excited that we have Amy Janik with us to talk about damselflies and dragonflies. And I just want to remind you that next week is the last one in our series, and that is Dr. Michael Ward, and he's going to talk about excuse me, making your yard and garden home for birds. And I think that it'll be another one in the series that will be really knock it out of the ballpark. So I'm going to let Amy take over and we'll have a time. Amy, do you want to ask questions as we go or do you want to wait till the end?
Probably till the end, if that's okay.
Okay, that's fine. And I'll keep an eye on the chats, and so we can have those at the end. And so take it away, Amy. Thank you so much.
Yeah, thank you, Holly. Hi, everyone. My name is Amy Janik. I'm a research specialist at the National Great Rivers Research and Education Center. We also call it NGREC. We're down in Alton, Illinois, and we do a lot of research on anything that is impacted or affected by the floodplains of the Mississippi River. And so my research that I've been focusing on and I'm a co-principal investigator on is our Dragonflies and Damselflies project. And so I'm going to kind of just give you a little bit of a rundown of what we've been doing at NGREC. Let me see if I can move the next slide. Yeah, so what I'm going to go through today is I'm going to kind of give you all some information about the life history and background of odonates, the conservation needs that they have, especially in the state of Illinois, the work that we're doing at NGREC right now and what I'm focusing on, and the objectives and methods of our projects, and then what we're looking forward to do in the next year or in future years as well. So to start, the order of Odonata is made up of two suborders. We have the Anisoptera, which are dragonflies, and Zygoptera, which are damselflies. You'll hear me refer to them as odonates collectively throughout the presentation. The adults are easily recognizable, as I'm sure you all know, with dragonflies being fairly robust and perch with their wings open, and damselflies being slender and most species perch with their wings closed. So dragonflies and damselflies can be characterized by their rather large eyes, their tiny antennae, chewing mouthparts, and well-developed wing muscles. They are carnivorous at all stages of their life cycle. There have been over 6,000 species described in the world, and they occur on every continent except for Antarctica. They can be found in a variety of freshwater habitats, ranging from ephemeral streams to large lakes. And so odonates first appear in the fossil record about 250 million years ago. Although some groups and suborders have long gone extinct, there are many families that we recognize today that were present at the start of the Jurassic period. They appear before the dinosaurs and long outlast them. The odonate life cycle spans aquatic and terrestrial ecosystems where they play key roles in their environments. So unlike butterflies, odonates have an incomplete metamorphosis, which means that they lack a pupa or a chrysalis stage between their larva and their adult. The odonate nymphs actually will stay active in the water while they're developing, and then they molt from their larval skin, very similar to what cicadas do. And so I'm going to kind of take you through each of their life cycle stages. I'm going to focus first on their egg stage. There are a variety of ways that female odonates oviposit or lay eggs. Some species will directly put them into the water. Some will just drop them from the air into the water. Other species will lay them onto or into aquatic plants. And adult females can lay a clutch of eggs daily. Each clutch could range anywhere from 100 to 1,000 eggs, depending on the species. The developing eggs can hatch in a matter of 5 days or 250 days based on the environmental conditions of the ecosystem they inhabit. There's clearly a lot of variation among these species, especially for egg development. Next, we have the nymph or the larva stage. I'm going to guess that this is the stage that we're one of the most least familiar with. Odonates will spend actually most of their life underwater in this nymph stage. The variation in development time is also quite extreme. In warmer temperatures, species can complete larval development in a month, but in colder, high latitude streams, larvae can take up to 8 years to fully develop. So before moving on to the adult stage, I wanted to give a quick background on the body plan and some really neat morphological characteristics of odonates. So nymphs and adults both have that typical insect 3-part body plan. We have the head, the thorax, and the abdomen. One of the more fascinating aspects of an odonate nymph specifically is in the head. It's the labium, or essentially the lower lip of the mouth part. Depending on the species, the labium can be flat or scoop-shaped, more like a spoon. It is one of the key characteristics used to identify which family the nymphs belong to. The labium actually allows for rapid strike when hunting for prey, essentially acting as a catapult with stored kinetic energy, and it startles and then captures its prey. They have these needle-like projections at the end of their labium called the movable hook, which is also used to keep a hold and stab at prey. And you can get a really good look on the damselfly larva with its movable hooks at the end of its labium. So here's a few examples of nymphs that we have in our lab right now that we collected during our pilot season last year. Using a digital microscope with a camera, I was able to take these images. We actually just recently got a new microscope that can take a lot better images, so I'm excited to start using that. The length and shape of the mouth part is what helps me narrow down what families an individual might belong to, as well as the presence of any of the setes, which are those movable setae and movable hooks. And those setes are structures that resemble hairs or bristles, and that helps to kind of keep things from escaping from their mouth parts as well. So for the adult head, we have the most obvious part is clearly the large prominent eyes. Dragonfly eyes are close together and touching, whereas damselflies are spread apart and wide. Vision is the primary sense used by odonates, and in most species, the compound eyes can contain up to 30,000 simple eyes, each functioning independently of one another. One of the many reasons odonate adults are so hard to catch in the field is due to their ability to rotate their head 170 degrees, and they can also rotate their head upwards 90 degrees. They lack many blind spots, and it makes them difficult for researchers like me to try and catch them with a net. So the thorax in adults and nymphs serve essentially the same purpose. The thorax is where the legs and wings, or the wing pads for nymphs, are located. The wings of adults are constructed with a complex set of veins that support them, that support incredibly thin membrane. They're made of a flexible protein that allows the wings to bend and move and then recover. The forewings operate independently of one another, allowing for optimal flight performance and increased agility. The average flight speed of a medium-sized dragonfly is about 4.5 miles per hour. However, there are some skimmers that can reach up to 9 miles per hour during territorial defense. So next, then we have the abdomen, which kind of makes up the most of the odonate body plan in both the nymphs and the adults. Dragonfly nymph abdomens can range from stout and robust, similar to like a disc, or elongate and cylindrical. The damselfly nymphs are much more slender and narrow, with prominent gills resembling leaf-like appendages at the end of their abdomen. So the nymph abdomen is one of the cooler characteristics of odonates. This is where their gills are located for nymphs to breathe. For damselflies, like I said, those are the leaf-like appendages at the end of their body. However, for dragonflies, their rectum contains their respiratory system. So they essentially breathe through their butt. Because of their strong diaphragm in the abdomen, they can water can be expelled very rapidly from the rectum, which will then shoot the nymph forward through jet propulsion, allowing them to escape from any predators and from anything that's trying to eat it, and then also helps when they're trying to attack predators or attack prey. So while some nymphs may live underwater for a few years, the lifespan of an adult odonate is much shorter. Adult dragonflies and damselflies can live between 1 month and a few months, during which they spend their time feeding, basking in the sun, and reproducing. The diet of odonate nymphs can vary from large aquatic organisms to small aquatic insects, essentially anything that can really fit in their mouth when they're underwater. Larger or more developed dragonfly nymphs can forage on small fish or tadpoles, whereas small dragonfly nymphs and damselflies can eat things like mosquito larvae, which is very helpful, or mayfly nymphs. They'll also eat each other, and they're also very successful predators due to their ability to camouflage within any of the detritus or muck of in a stream or a pond, and they can swim really quickly through vegetation when hunting, especially with that jet propulsion. Adult odonates prey can range also greatly. We do tiny insects can make up much of the dragonfly and damselfly diet, but dragonflies and damselflies, butterflies, and even hummingbirds have also become prey for some of the largest dragonfly species. They are incredibly impressive and successful predators if you've ever watched them. So odonates have a couple of different ways to stay away from predators, because they do have some. Some of the female odonates are cryptic and able to blend in with their surroundings to avoid being eaten, but also using their speed and agility, odonates use a zigzag flight pattern to outsmart their predator while being chased. However, there are, of course, a couple unlucky ones that will still become prey to predators such as birds, bats, frogs, and lizards. So odonates are a very colorful group of insects representing all the colors of the rainbow, essentially. These vibrant colors are not just for aesthetics, they can serve specific purposes. Males are commonly the more colorful ones to make it easier not to confuse them with females when selecting a mate. Odonates living in sunny wetlands will have brighter colors to be able to reflect solar rays to regulate their temperatures. And then ones, especially like the fawn darner, is a much more darker brown one who likes to live in a shady stream, is much darker to absorb that heat that it does have. Certain colors are handy camouflage that help the dragonflies blend into their surroundings and stay safe. But they can also be used to attract mates or to warn off predators, kind of similar to birds. So mating usually takes place near water, where some male dragonflies will come migrate early, and they'll set up territories to prevent too many other males from being in the area and increasing any competition. So if you see a lot of male dragonflies chasing each other, that's usually what's going on. Courtship varies among odonate families. Many damselfly courtships can be very elaborate and flashy, which is also very similar to courtship displays in birds. So when dragonflies and damselflies mate, the pairs can form a wheel position. Not all do, but some can. And this kind of wheel position is unique among insects. Quickly after mating, females will oviposit into the water either by themselves or sometimes still in tandem with the male. So adults have specific flight seasons, which is a period when they are on the wing. The timing of a flight season is mostly based on larval development and environmental conditions. Some species will have a synchronized emergence and a synchronized die-off. Others will continue to emerge and have a flight season through the summer and fall. Many odonate nymphs will hibernate in the winter in the water and then emerge as an adult in the early spring. However, it's really rare for an adult to hibernate over the winter. So dragonflies can disperse and migrate long distances due to their strong, powerful flight. Damselflies also can disperse far distances, but they do it by flying vertical and into prevailing winds that then carry them. Species that their nymphs develop in ephemeral wetlands or wetlands that are going to be temporary and streams as well can fly far distances to find a new wetland as soon as they've emerged. So the best known migrant dragonfly in North America is the common green darner. It's definitely one of our larger dragonfly that we have in the States. In one study, the researchers attached like a tiny radio transmitter to the thorax of an adult common green darner and they tracked their movements during the fall migration on the East Coast. They found that they migrated about every 3 days during the daytime when wind speeds were favorable. They were able to travel about 36 miles in 6 days. Actually, currently right now, we have the common green darner migration going on in the Midwest. So if you live near a shore or the Great Lakes, you may notice these large amounts of these kind of greenish and blue with a blue abdomen dragonfly. They're very large. They're in large numbers. And they kind of do a similar migration as monarch butterflies, the multi-generation. And so if they had emerged as an adult recently, they don't have enough time to let their eggs and everything develop in the water. And so they start to move south. They can get to Mexico, Central America, and they try to find areas where they can oviposit their eggs in areas that will be warm enough for them. So I'd keep an eye out for that. I believe it'll go on until about mid-October. It's really cool to see. So since odonates span both aquatic and terrestrial habitats, they are vulnerable to threats that impact both freshwater ecosystems and landscapes. Some of the challenges dragonflies and damselflies face are habitat loss, pollution, and climate change. Wetlands serve as one of the world's most economically very valuable ecosystem, and they're disappearing really fast. Climate change, which is already a threat to many insect species, will just continue to be more extreme, especially more extreme droughts. And then that will clearly impact odonates and decimate their populations, because if they don't have fresh water, they can't feed, they can't lay their eggs, they can't mate. So some species of dragonflies and damselflies are generalists, which mean that they can eat or inhabit various areas, which could allow them to adapt to more urban environments, which I'm sure you've seen plenty of odonates in more urban areas. However, there are many specialist species that you haven't seen in these urban areas that are really focused on these kind of more pristine habitats. And they're maybe focused a little bit more on a certain type of prey or need a specific plant to lay their eggs. These species will definitely not thrive in urban areas. So even the odonate species that can adapt to more urban habitats will still be sensitive to water quality. We have various pollutants such as road salt, heavy metals, and insecticides that can be detrimental to both nymph and adult populations. So due to the many environmental threats to odonates and the key functions that they play in ecosystems, it's crucial to assess the status of odonates in Illinois. Conservation of invertebrates or insects is often neglected, and many species in need are never really recognized due to a lack of information and baseline data. So in Illinois, we lack data on distributions, rarity, and vulnerability of many families of odonates. It's hard to kind of figure out how to protect an order of insects without this knowledge of where they're occurring. So this is a heat map that I got from Odonata Central when just looking at the state of Illinois. And so it shows the records that are collected through community scientists. The red and orange areas are where there are over 30 records of a species. All the purple is less than that, and no color means no data. So clearly the Chicagoland area is kind of the spot where we have some consistent records for odonates. However, there is still a lot more of the state to explore. And so our focus is, to conduct an in-depth assessment of that conservation status of these other taxon groups at the state level. And this way we can kind of help mitigate this lack of conservation management for invertebrates, especially since they are in decline. So here's now getting kind of more into the specifics of our project at NGRAC, which is funded through an Illinois Department of Natural Resources grant. So right now our two objectives are to design a protocol that improves sampling and surveying methods for both life stages of odonates. It'd be really great to have a standardized protocol for state parks and community parks and things like that to be able to follow. That way we can use that to keep track of different odonates in different locations. And then using that information, we're going to utilize occupancy and detection models to determine species distributions and then what factors influence species occurrences in Illinois specifically. And so I'm going to kind of walk through some of the steps that we're taking to achieve these objectives. So to start, we want to fill in these gaps of knowledge and have, so far we've been assembling and analyzing data from insect collections in Illinois natural history museums. By doing this, we are able to assess what species of odonates are underrepresented. We have some really cool historical data that can show us the first time something was spotted in the state, and hopefully has a GPS location as well that goes along with it. We compile this, and then we also will then, not only from natural history museums, we're also using data from verified naturalist databases such as iNaturalist and Odonata Central. So then based off of those historical records and geospatial data, we can identify watersheds and habitats that are undersampled and contain species that may be declining or rare. So like I had said, we know that kind of the Chicago metropolitan area is definitely heavily sampled. It doesn't necessarily mean that there's more odonates there. It's just there's more people there that are able to report them. And so this is a map of the watersheds of Illinois that the Illinois River Watch program at NGRAC utilizes for their different field sampling. If some of you are volunteers for that, this may look familiar. So to kind of keep things standardized, for the Odonate project, we also use this map so that we can select sites from various watersheds in the state. And then we can also compare our data to some of the River Watch's data as well. So then once we do that, we're going to create a targeted field assessment to kind of finish filling in these knowledge gaps. We're going to target specific areas of Illinois with wetlands and streams, so ponds, floodplain marshes. We do larger streams. We also do the Sangamon River, so a whole bunch of different spots, kind of wherever we can get some access to and get good data to, and it is safe for us to access. So then we compile this information from the field so that we can make suggestions to state and regional wildlife management plans. So our odonate sampling takes place during the peak flight period. So that would be like April to October is kind of like the longest one where we can kind of hit quite a few species flight periods. There's usually 2 researchers that go out, me and another field technician. We sample at 5 to 6 sites per region in a combination of wetland and stream habitats. We do kind of, in order to verify an occupancy model, we have to do 3 closed sampling rounds that last 4 weeks, so 1 region per week. And we hit 4 different regions of the state that we kind of separated ourselves based off of first freezes of the season. And then we will actually, those 5 and 6 sites per region, we will sample 3 times in 1 week. So
Wait,
for
what?
Just listen to your thing and forget the package.
Yeah, but that makes me really mad.
Well, honey, I'm
sorry about that. So we had for our 2023 field season, we sampled from 23 sites in undersampled regions of Illinois. We had 5 to 6 sites in regions such as Eastern Shawnee National Forest, Grafton, Illinois, Champaign, Illinois, and Rockford, Illinois. We would travel about an hour outside of these areas, but those were kind of the main home bases that we would be at. We kind of focused our sampling sites at properties such as state parks, county forest preserves, and Army Corps properties. Up for those of you around the Champaign area, we were at Lake of the Woods and the Sangamon River Forest Preserve, Kickapoo State Park, which were really beautiful. So specifically, one of my main focuses of this Odonate project is to make sure that we're designing a sampling protocol for these field assessments, and one that can be easy enough to then distribute to everyday people, state parks, things like that, to hopefully try and attempt to continue to collect data. And so in order to know what species are occupying stream and pond habitats, we need to have a standardized method for collecting from different locations and also for different species. And so I'm going to run through some of those methods that we used for our field season this year. And before collecting specimens, one of the most important parts is that we take a variety of data that included habitat and environmental data. So unless it was like actively storming, we would try to sample during all weather types. That way we weren't being biased to any specific species that really prefer sunny, warm days. Some species do prefer the more shady and cloud covery days. So even when it was raining, we would still be out there looking for odonates. All of our environmental and habitat data is then going to be used for our future models that will help us determine when and what conditions certain odonates might prefer. So the most important equipment that we use are our nets. We use butterfly nets or aerial nets to catch the adult odonates. We go through a lot of butterfly nets. It's much more aggressive trying to catch odonates than it is butterflies. And so we break them easily, unfortunately. But we then use also aquatic D-frame dip nets that have a very fine mesh to collect the odonate nymphs. And so these nets are much sturdier due to needing to scrape them along rocks, logs, and stream banks to find the nymphs. So I'm first going to go over our aquatic sampling protocol. So like I had said, we have 2 researchers that are usually out there in the field. We sample along, we each sample along our own 25-meter transect that connect to each other to look for dragonfly and damselfly nymphs. So for streams specifically, our transects are essentially a straight line through the stream bed. If it's more of a river and we can't get to the middle of the stream, then we try and focus on the stream banks. In ponds and wetlands, it's a little bit more difficult, especially as they dry up. But we usually, to avoid getting really stuck in the muck, we kind of do a circular transect along the shoreline of the wetland. And the point of this transect is essentially to help us ensure we are sampling from the same amount of habitat at each site. And also to kind of give us a time limit so we're not spending all day at one site trying to sample everything. So within our 25-meter transects, we will spend time sampling from the best microhabitats within a stream or pond. These microhabitats are areas where odonate nymphs really like to hide and burrow and will give us the best chance to finding them. It's important to spend time at different microhabitats within our transect as different species utilize different parts of a stream. So next, in both ponds and streams, we look for leaf packs, particularly decaying leaf packs. The nymphs will prefer to hide out in leaf packs that have been in the water for a while, as that likes to attract a lot of their prey species.
Yeah, I'm sitting down.
So undercut banks and streams, and then the banks of a pond are also really important. A lot of roots from the surrounding vegetation can be found in this area. And damselfly nymphs like to attach themselves to those along with fallen branches or snags that have been sitting in the water for a while. So aquatic sampling kind of involves a lot of kicking up sediment, scraping rocks and roots to force the nymphs to come out of their hiding spots and into the net. The dip net is usually set up downstream, so that way if any nymphs kind of get caught in the kind of like the water column, it'll flow then into the net. That's usually the goal. So when dip netting, you pull up a lot of other things like sediment, mud, leaves, rocks, and lots of other tiny insects or aquatic organisms. You have to just kind of sort through all of that to look specifically for dragonfly and damselflies. Dragonflies are relatively distinguishable due to their kind of chunky state, but damselflies are much more discreet and can kind of look similar to other macroinvertebrates such as mayfly nymphs. One of the really useful tips when looking for damselfly nymphs is they have a tendency to kind of raise their back of their abdomen up in the air like a scorpion. That kind of helps them stick out. Otherwise, they kind of just look like a bunch of sticks. But we also tend to find some other really cool and neat organisms, such as crayfish, or a couple weeks ago, I caught a small soft shell spiny turtle baby in my net, which was very exciting. And we also keep note of the other things that we find as they might kind of influence what populations of odonates we see. And so aerial sampling, although I wish it was, it's not quite the idyllic activity that you'd imagine with butterfly catching. It is relatively the same process, but much more intense. They have that zigzag pattern that I was telling you about earlier, that they also use on humans, and it's very effective, so it makes it very hard for me to catch them. But we're also chasing them through in muck boots through wet rocks and sticky muck, and it's very challenging to say the least. Have gotten a lot better at it though by the end of the field season, which has been very exciting. So following that same transect as the aquatic, we walk our areas again, focusing on favorite perching locations of odonates. So we're sometimes going to focus on sunny spots or sticks or branches in the water or on vegetation. However, not all species like to perch, and some stay flying while they are active. So at the same time, we are attempting to catch anything flying around or above our head, which takes a lot of special skills. One positive is dragonflies especially have a tendency to kind of utilize that same flight pattern. So you can hope that if you see them come by once, you can hope that they're going to come by kind of in that similar area. They also like to use very similar perches, which are some things that we've learned while being in the field with them. So then after we do our both collecting periods, we'll then perform a visual survey of adults that are on the wing within that same transect in order to include any species that were really hard to catch or we just didn't observe during the aerial and they just appeared during the visual. And so to do this, it's usually just me who does that, while my other field tech will be doing some environmental variable collection. But I will use either a scope or binoculars in order to get the best view to ID them in the field. So once we're done, we collect voucher specimens, which just means that we're collecting only one individual from a species at each site as to not over collect from a population. But voucher specimens are really important as they allow us to verify the species presence. But then they can also be preserved for any future research needs, such as if we want to do DNA, anything like that, then we have a specimen. So for nymphs, we try to follow kind of a similar process. However, it's much harder to ID nymphs in the field because a microscope is needed, as it's usually very small characteristics such as their mouth parts or their eye size, things like that. But we will then take these specimens back to the NGRAC field station where we can take a closer look and ID them in the lab. Once we are done with identification and most of our data collection, we'll preserve the specimens. Our plan is to have the specimens sent to the INHS museum collection and to have a collection here at NGRAC for our education program. So back in the lab, we try to break down the closest ID that we can using various resources, especially a dichotomous key, which is a scientific method to identify different organisms based on observable traits. So the ID process starts at the largest taxonomic unit. So for us, it would be the suborder. Is it a dragonfly or is it a damselfly? Pretty easy to answer that. But then we continue to move through to families, genus, and then to species. So based on preservation or size of the specimen, we might not make it down to the species, but we try to get to as close as we can to best represent the data that's collected. And here's kind of a setup of how I have it for both nymphs and for adults. And on the right is the very fancy new microscope and camera that we got that just really helps to get a better look at some of these really neat parts of the odonates. So this kind of essentially leads us to where we are right now at NGRAC. Actually just finished up the last round of our odonate field season last week in Rockford. And so we have one more year of funding for this project. And so for the winter and next year, we will be confirming the identity of collected specimens for both the adult and the larval odonates. So just like the pictures from the last slide, we'll be spending a lot of time with the microscopes to kind of just confirm if we had any identifications in the field, just to make sure that we were correct. And then we will, starting in next spring, we will then continue our field protocol for at least 25 new sites in 2024 to continue to fill gaps in the state of Illinois. Hopefully we can hit more sites other than 25, but that's at least our goal. And then lastly, at the end, we'll have 2 years of data collected, hopefully have analysis completed, and then our plan is to then disseminate this odonate distribution data in the state of Illinois to inform our state partners, which is the specifically the Illinois Department of Natural Resources. So that way, if we are realizing that we have some more rare species in Illinois than we thought, or that species are declining, we can kind of set up a protocol that state parks and other land managers can potentially start, managing for these species, but also just monitoring as well regularly. And I know we already have some odonate surveys that go on, especially in the Chicago area, and I think down near Shawnee as well. And so kind of hoping to get programs like that to continue so that we can keep an eye on these species. And it would also be great to look at different life cycles, not just adults, to also focus on those nymphs as they're really important. So that is my talk about odonates. And so I want to thank you for listening, and I hope that was useful, and I hope you learned something new today. But if there's any questions, I'm happy to take them. And if you think of something later, there's my name and my email address for my work. But yeah, I can take any questions if you all have them.
Oh, Christopher Carlson asked, how many species have you identified?
Oh, wow. I was just adding this up the other day. I think I'm at about 80 different species. No, probably about 50 species collected. That's including dragonflies and damselflies, and we have some repeats. But I think in total we have almost 200 individuals collected so far from this field season. But I'm at about 50 different species that I've identified, but still kind of going through the data as we had just finished last week.
And then he also asks, how many species could I expect to see at my backyard pond in Urbana?
Oh, that's a great question. So especially at wetlands and ponds are hotspots for sure. You can probably find about, especially in the middle of June and July, you can probably find about 20 different species. You'll have a lot of variety of damselflies in the vegetation, but then flying over on a really good day, you could get about 20. The dragonflies will be flying over, and you can have they don't get too territorial with each other. They kind of let each other be there because they'll utilize different areas. But yeah, I would say about 20 on a really good day, maybe even 15 on a regular day.
What can we do to increase them in our own backyards? Is there just a water source? Is there certain kind of plants that they enjoy? How can we help?
Yeah, that's a great question. So water sources for sure. If they need that in order to mate and also feed. Native plants there's really nothing specific that they like to use because they're not pollinators, but they eat a lot of pollinators. And so, bringing them to your yard where if you have a lot of native plants, you're going to get a lot of those other native insects and things like that, that will definitely bring them. They're really important. It's good to have them because if you also have a water source, you're most likely going to have mosquito larvae, things like that. They're really great at controlling those types of things.
I think you said that there was, I'm not sure if it was a damselfly, it took like 8 or 15 years or something to develop. Did you say that?
8 years. Yeah. 8 years in the water. So those live at like higher latitudes in like a mountain stream or something like that. They can take up to 8 years to develop because usually a nymph will kind of go through different molts throughout its time, even in the water. And it needs kind of the correct water temperatures and things like that to do that. And they can't if it's just kind of staying at very cold temperatures. So some nymphs will take about 8 years underwater to develop, which is pretty crazy.
I just can't imagine that they aren't eaten up by that time.
It's
by something or hit by a car. I mean, I just can't believe that that you have very many after that many years.
Yeah, my thought is that if it's that hard to live in, there may not be that many predators of them also inhabiting that stream.
My goodness.
Yeah.
Other questions? Amy, you did a great job. And
Thank you.
Karen Horn wrote, thanks so much for the seminar. It was great information. Honestly, I did not have a lot of knowledge about the topic when we started this evening, but I think I'll be paying a lot more attention now that I know I really didn't know there was so many different species here in Illinois.
Yeah, and it's kind of tricky to see them always if they're not under the correct light setting or anything like that. But yeah, if you kind of look closely, especially even in urban areas, we'll get the common green darners. And even I'm in St. Louis and I saw a wandering glider the other day right by the arch. So there's really a lot there and it's really cool.
Now we have one more that said, under normal non-migrating tones, how high do they fly?
That's a good question. They don't fly very high. So a lot of the darner species and some skimmer species, the bigger ones, will fly and actually sit at the top of trees. So that's about 50 feet is probably the max. Otherwise, damselflies are almost always only a couple of feet off the ground, other than maybe ebony jewel wings that have that thicker wing. But other dragonflies that are smaller kind of stay pretty low, only a couple feet off of a pond or a stream. But I have seen, especially like emeralds and the darners and skimmers I've actually seen them sitting at the top of a tree that's about 30 to 40 feet high.
Were you always interested in this topic, or was this just something that you got into in college?
Actually my background is more in birds, but I've kind of just as like a side activity, I've learned how cool dragonflies was, and I started to recognize how many species just like birds. And so it's kind of a very similar hobby. And have just, as I've been learning more and more about them, I've just become more and more fascinated. They're just such cool species. So this is kind of a more newer phase, but I've been really enjoying it.
Well, you would like next week when we have Dr. Michael Ward on. Have you heard him speak before?
I haven't, but I worked with him a bit for my master's program on the blue-winged and golden-winged warbler. So he's a great person.
Mm-hmm. We will. If you get a chance, you should join us next week because he will do a nice job. Anybody else have questions before Amy gets away from us? Okay, what'll have this? If you think of something in the next, tomorrow you wake up in the middle of the night and you're like, oh my gosh, I forgot to ask Amy something really important about this. You can either contact me and I will send on any questions to Amy, or you can contact Amy directly and we'll try and get those questions answered. And again, I want to remind you that we will meet again next Wednesday for the last one in our Wilderness Wednesday series. Say that 10 times fast. And we have Dr. Ward. And that will be the wrap up for the September series. And then we'll do another series in January of Wilderness Wednesdays again. So if you have any questions, let myself or Amy know. And we were so excited to have everybody on. Thank you so much. Thank you, Amy.
Yeah, thank you so much for inviting me.